All right, well, welcome to San Francisco. Those of you who just got in, some people have been here a while. Thanks for coming to our launch of S2114. And I'll just, Jonathan Friedberg, I chair the lymphoma committee. And before I introduce Brian, I want to just frame this study as far as the importance of this in the whole landscape of diffuse large B-cell lymphoma. So I think arguably one of the biggest advances that we've seen in that disease over the last decade has been CAR T-cell therapy, which has gone literally from a few patients getting it in phase 1 to 3 approved agents being used rather routinely now, and largely for many patients replacing autologous stem cell transplant as the salvage option of choice. And that's based on incredibly promising data suggesting that this is curative therapy at a higher rate than what we used to see. But there's still problems with CAR T cells. And the bottom line is that even in the clinical trial populations, the best, most selected patient populations, the majority of patients that get this treatment will not be cured. So this is the first NCTN effort in CAR T-cell treatment. And what's great about this study is it leverages commercial CAR T-cell treatment. So these are patients, quote unquote, real world patients that are going and getting their CAR T-cell treatment. And we're adding to the commercial CAR T-cell treatment in a select group of patients who we feel are at the highest risk of recurrence to try to avoid that recurrence. So this study's been designed specifically to try to make it easy to accrue. And if you consider the number of patients that are getting commercial CAR T-cell products now in the United States, I think we're optimistic that this study will accrue very well and inform our community and hopefully pave the way for a phase 3 trial. But it will also start to collect data within the NCTN for patients getting this treatment. And I think that's going to be very valuable as a benchmark and to understand who these patients really are in a cooperative group setting. So with that, I'm going to introduce Brian Hess, who has helped lead a broad team of individuals who are listed on this slide to design and execute this study. And I believe he told me that he's enrolled the first patient on the study as well. So his hard work continues. We want this to be interactive as well. So if there are questions along the way, please interrupt.
Great, thank you, Dr. Friedberg. Really great to finally have this, this study activated. We really hope to open this in a lot of centers, not only at SWOG centers, but ECOG and Alliance. And we've had a lot of help from representatives from those groups as well. So hopefully we continue that collaboration, but really excited to finally kick this off today. So Dr. Friedberg did a really good job of framing the question here. I put the schema up just for a quick orientation. For all patients, there will be two-step registration. So patients will undergo first registration prior to lymphodepleting chemotherapy and CAR T. All of these patients will have a day 30 PET, which will be centrally reviewed. For patients with complete remission and progressive disease, they will not be eligible for randomization. Patients with stable disease and PR on that central review will be eligible for randomization to either Mosin alone, Pola alone, the combination, or observation. And so this is the group that we have deemed as high risk for relapse that we're trying to reduce that risk. If patients are randomized to observation, they will have the option to cross over to the combination of Mosin and Pola if they have definitive progression within 1 year of randomizing. So a little bit of background that Dr. Friedberg already gave really nicely. Diffuse large B-cell lymphoma, the most common lymphoma that we see in clinic, close to 27,000 patients in the US diagnosed per year. And the majority of these patients are actually cured with frontline therapy, but there's still a large group that are either refractory or relapse after that frontline therapy. And with the second line approval of CD19 CAR T, CD19 CAR T is now the standard for nearly all patients with relapsed large cell lymphoma. And this number right here, as Dr. Friedberg alluded to, is really why we are developing this study, the 60 to 65%. We know CD19 CAR T has been an incredible breakthrough. Certainly nobody's denying that. It's really changed the landscape of how we treat relapsed large cell lymphoma. But still, the majority of these patients receiving CD19 CAR T are relapsing. So we asked, how do we identify these patients at high risk for relapse post CD19 CAR T? And we really think this day 30 PET gives us a good mechanism for this. You can see about 50% of patients have a complete remission at this day 30 PET. About 20% will have progressive disease or progress prior to that day 30 PET. And then about a third of patients have stable disease or partial remission. Not surprisingly, if you have a complete remission at that day 30 PET, you're more likely to have a durable response. About 2/3 of these patients have a durable response. And if you're in the stable disease PR group, the majority of these patients will end up relapsing. So only about 25 to 30% of these patients have a durable response. So this is the patient population we deemed as high risk for relapse. And then the next step was, if we think these patients are high risk for relapse post-CAR T with stable disease and PR, what therapies could we give that would be not only effective, but also safe, given that they'll be received in such close proximity to CAR T? And a lot of the traditional therapies that we have been given post-CAR T, like cytotoxic chemotherapy, really haven't been very promising at all. So we turn towards more novel therapies like mosunetuzumab, which is a CD20-directed BiTE, has a lot of promise in phase 2 trials in relapsed large cell lymphoma, also has the potential to augment persisting CAR T cells in the context of this study. And then polatuzumab is an antibody drug conjugate to CD79B on B cells. It has approvals in conjunction with other therapies, both in the frontline setting and in the relapse setting as well. And when these therapies have been given together in relapsed large cell lymphoma, they've shown a lot of promise with a complete remission rates nearly 50% in a high-risk patient population. And these are patients with florid relapse. And what's also interesting is patients that in this study that received prior CAR T, those response rates were consistent. So the primary objective for this study became comparing giving consolidation therapy with either mosun, pola, the combination versus observation in these patients with stable disease or PR at that day 30 PET, where we'll be trying to improve the 1-year progression-free survival of 25%, which is estimated for this observation group to 50% or greater with each of these consolidation arms. Secondary objectives include looking at overall survival and CR conversion rate for each of these consolidation arms versus control, looking at adverse events within each of these consolidation arms. We'll also be collecting and banking these day 30 PETs, so we'll be able to look at total metabolic tumor volume, SUV max, some product of diameters, and how that correlates to CR conversion in each of these consolidation arms as well as control. We'll also be banking these PET scans prior to CAR T, as well as peripheral blood draws for specifically for minimal residual disease. So again, just to orient, patients enrolling in step 1 can enroll anytime after they consent for CAR T apheresis or for CAR T collection at the local center and up until lymphodepleting chemotherapy starts. And all patients registered to step 1 will have a day 30 PET scan, which is centrally reviewed. And for all patients registering to step 1, again, they are planned to receive one of these CD19 CAR T cell therapies that are FDA approved for large cell lymphoma for one of these indications listed here, whether it's 3rd line or greater, where all 3 of these constructs are approved, 2nd line when relapsing in less than 12 months for frontline therapy for axi-cel and liso-cel, or 2nd line for non-transplant candidates for liso-cel specifically. And there's a few logistical complexities with this study for sure. So I wanted to put this figure up for the study teams. just so we can make this as clear as possible. So again, patients sign up for first registration consent anytime after CAR T collection consent and up until lymphodepleting chemotherapy is started. And so when we identify these patients in clinic that are CAR T candidates, they undergo collection. It typically takes close to 3 weeks for these cells to be manufactured. In that time, patients may or may not get bridging therapy with radiation or systemic chemotherapy. And then once the cells are sent back, they receive lymphodepleting chemotherapy for either 2 or 3 days, and then get CAR T infusion after a couple of days of rest. So on this study, patients are allowed to receive bridging therapy, but cannot receive POLA or Mosun. LD chemotherapy needs to be started within 60 days of signing consent. Most patients are starting this within 30 or so days. So I don't see this as a big issue with patients. Patients typically will receive fludarabine and Cytoxan as conditioning therapy, but some will receive alternative regimens such as bendamustine at different centers. This pre-CAR T PET, again, will be done prior to lymphodepleting chemotherapy. It needs to be completed within 60 days of LD chemotherapy start. If patient has bridging therapy with radiation or systemic chemotherapy, that PET needs to be repeated. In terms of the product and infusion, sometimes the CAR T cell product does not meet the parameters set by the FDA. In these situations, patients can receive these CAR T cell products on single patient INDs or expanded access protocols. Those patients would not be allowed to continue on 2114, however. And then the product is infused 2 to 14 days after LD chemotherapy completion, which is standard of care. These post-CAR T PETs, which is the day 30 PET, again, which will be centrally reviewed, needs to be completed within 25 to 40 days from CAR T infusion. So these are some of the logistics that we just wanted to make clear to the teams. This is all included in the protocol as well as repeated in the eligibility criteria for step 1. And so for all of these centers that are treating with CAR T cell therapy, they need to be REMS certified for each of the products that they're using, and each provider needs to be REMS certified to provide these products. So this is not an extra step required by the study, but this is something that the FDA mandates for management of these CAR T cell patients. But before and after CAR T, we do include some general guidance in the protocol, but a lot of this is included in the SOPs for each individual institution. And a lot of the logistics regarding CAR T cell infusion, collection of CAR T cells, management of cytokine release syndrome or ICANS, this is all going to be per institutional guidelines, because we want to, these CAR T cell products are being given as standard of care. And we think, these centers that are giving them as standard of care really know how to manage these patients. So key eligibility for step 1. Patients need to have a diagnosis of diffuse large B-cell lymphoma, follicular lymphoma 3B, or primary mediastinal. You can have transformed disease, but only if it's from marginal zone lymphoma or follicular lymphoma. So we don't allow patients with Richter's transformation to enroll in the study. Patients can have evidence of CNS disease, but they cannot be symptomatic from that disease. So if a patient has an MRI, for instance, that shows leptomeningeal disease, they can be enrolled on first registration as long as they're not symptomatic from that disease. In terms of prior concurrent therapy, a lot of that was included in logistics of the previous slides. Again, need to be a candidate for an FDA-approved CD19 CAR for one of these diagnoses, can't have a history of previous CAR T infusion. Again, you can receive bridging therapy after CAR T cell collection. POLA and Mosun is excluded. PET-CT, again, needs to be repeated after bridging therapy. One important point I think to note is that patients can have received prior Mosun or polatuzumab. They just can't receive it as bridging therapy. And this is important because polatuzumab, as many of you know, was just approved in the frontline setting with RCHP. So if a patient would have received RCHP plus POLA and progressed, they would be allowed to enroll on this study as long as they did not progress within 6 months. In terms of clinical and laboratory criteria, we have parameters for age and performance status, organ function, all included in the protocol. Patients cannot have neuropathy grade 2 or greater because of the potential for receiving polatuzumab as consolidation. And then we do allow patients with history of Hep B, Hep C, or HIV with all those parameters described here, but each of these patients would need to have a negative viral load. So moving to step 2, again, patients with complete remission, these patients are not eligible for randomization. They would undergo observation with disease assessments and biomarker draws per protocol. Those disease assessments really will mirror standard of care, so we do not expect that these patients have any extra visits for this study. Same for patients with progressive disease. They will not be eligible for randomization and will receive physician-directed care, whether that be standard of care treatment or on another clinical trial. And these patients with stable disease or PR by central review will be eligible for step 2 randomization, again, to one of these arms, Mosin alone, Pola alone, the combination or observation, where there'll be 30 patients per arm. You see the stratifications here where we stratify by stable disease versus PR. On day 30, we know that patients with stable disease do have worse outcomes, and also stratifying by receiving CAR-T as second-line therapy versus third-line or greater. And again, patients randomized to observation will have the potential to cross over to the combination of Mosin and Pola if they have definitive progression within 1 year of randomization. So getting into the details of eligibility for step 2, we do repeat all these requirements in the protocol. Didn't want to repeat them here, but they need to fulfill all those steps for step 1. Again, that 1-month PET needs to be done within 25 to 40 days of CAR-T infusion and stable disease or PR by central review. If randomization is delayed greater than 21 days from that PET and a patient has evidence of progression of disease, progressive disease, we do ask that that PET scan be repeated so that those patients that have progressed are not included in step 2. If for some reason the central review response cannot be completed because of the quality of the PET, those patients are not eligible for randomization. And then one, I think, really important point here is that patients can be randomized up to 60 days after CAR-T infusion. And this is important because a lot of patients can have toxicity from LD chemotherapy and CAR-T at day 30 post-CAR-T, which could potentially exclude patients from step 2 randomization. So for instance, something like cytopenias at day 30, we can very often see those resolve over time in the coming weeks. So we want to allow those patients to still enroll on study. If a patient had CNS involvement in step 1, they need to be in a complete remission at step 2 and submit a form that attests to this. So if you had leptomeningeal disease on an MRI in step 1, you need to be in a CR. You need to have a repeat MRI that shows you're in CR from that leptomeningeal disease to move on to be randomized to step 2. Prior concurrent therapy, again, you need to fulfill all those requirements that we went over in terms of logistics of PET CTs and timing of LD chemo and bridging therapy. No lymphoma-directed therapy since CAR-T infusion, and no intent for radiation after randomization. Clinical lab criteria, again, stressing the bone marrow function here in cytopenias. Patients need to have a neutrophil count of 1,000 or greater, platelets of 75,000 or greater within 7 days prior to step 2 randomization. These patients can receive growth factor and platelet transfusions, but they can't receive them within 72 hours of that blood draw that qualified them for step 2. And then patients cannot have any grade of cytokine release syndrome or ICANS when randomized to step 2. They can be on a taper of steroids as part of treatment of CRS or ICANS, but can't be any greater than 10 milligrams of prednisone or equivalent. So for the step 3 crossover, again, patients are eligible for step 3 crossover if they were randomized to observation and have had definitive progression within 1 year of randomization. Patients will have to have this definitive progression noted on imaging compared to day 30 PET. And again, that will need to be submitted to SWOG before these patients are registered. They must register within 28 days of the progression and not receive any lymphoma-directed therapy after progression. And a lot of the clinical and laboratory criteria reflects what we have in step 2 randomization. So in terms of the treatment arms, each of the treatment arms is providing time-limited therapy for these patients. You can see in arm 1 with Mosin, there's step-up dosing in cycle 1, and then they'll receive on day 1 of the subsequent cycles 2 through 8. Currently, what we have in the protocol is that patients will receive Dex pre-medication in cycle 1, and then it's optional for patients in cycle 2 and beyond. And it's optional to receive Tylenol and Benadryl. In the first revision, we will be submitting an amendment that likely makes dexamethasone and Tylenol and Benadryl mandated in cycles 1 and 2, and this will match up with the investigational brochure for Mosin. If patients have cytokine release syndrome with a prior administration of Mosin, the next administration, they will require a Dex pre-med until they don't have any further CRS events observed. And then if patients have grade 3 or 4 CRS with CAR T cell therapy, which really is happening nowadays in less than 5% of patients, they'll need to be admitted for observation for that first dose of Mosin for 24 hours. But outside of that, there's no mandated admission for observation for any of these patients in step-up dosing of Mosin. For POLA, it's given on day 1 of cycles 1 through 6 with Tylenol and Benadryl mandated as pre-meds. And for arm 3, where both of these drugs are given together, again, we see the step-up dosing with Mosin in cycle 1, and then polatuzumab is introduced on day 1 of cycle 2, and again, given for a total of 6 cycles. Polatuzumab will be infused first on each day that they'll be given together, and Pola and Mosin need to be given on day 1 of each cycle. I won't go over the steroid pre-meds for Mosin. Those will reflect what we discussed in arm 1. And the same for grade 3 CRS and admission for observation. So in terms of supportive care, growth factor is optional for patients in cycle 1 if they're randomized to one of those treatment arms, arms 1 to 3. And then if grade 3 or 4 neutropenia is subsequently noted, growth factor is mandated for all those subsequent cycles. Of course, we prohibit any anti-lymphoma-directed therapy. We prohibit steroids unless they're used as pre-meds or unless they're used as physiologic replacement. Live vaccines are not allowed up to 1 year post-CAR T. And if you're randomized to consolidation therapy, that's pushed back to 1 year after the last dose of consolidation therapy. And then in the protocol, we also include warnings regarding interactions with these consolidation therapies with other meds. So the disease assessments included all the disease assessments because we'll be having a schedule for all patients registered to step 1 with, again, that first disease assessment needed is that pre-lymphodepleting chemotherapy PET. Again, that needs to be done within 60 days of starting lymphodepleting chemotherapy, and repeated if bridging therapy is given. And again, all patients will get that day 30 PET. This, again, needs to be done within 25 to 40 days of CAR T infusion, centrally reviewed compared to the pre-CAR T PET. And then from there, the disease assessments are based on whether or not you're randomized to step 2. So patients with partial remission or stable disease that are randomized, their disease assessments are based on that randomization, whereas patients with complete remission, those disease assessments are relative to CAR T infusion. And just to note, one of the nuances that we include in the protocol for the patients that are randomized to partial remission and stable disease, that first PET that's done after randomization, we hope to have done just before cycle 4, day 1 for arms 1 to 3. And then one concern that we had mandating these PET scans for all these patients is making sure that, PET scans would potentially be approved for all these patients, and we didn't think that we could be sure of that. So starting at day 180, whether it be post-randomization or post-CAR T, we allow CTs with contrast to be done instead of PET scans, as long as the previous PET scan showed complete remission. So I think these are the noteworthy adverse events for the study teams to know for each of these consolidation therapies, especially considering they'll be given in such close proximity to CAR T again. And then in terms of the serious adverse events, these will require reporting regardless if they're attributed to drugs, and these are all listed in the protocol. So listed here is the table for dose modifications. And in terms of dose interruptions, if drugs are held for greater than 3 or up to 28 days, we consider that treatment delayed. And the reason for delay must be documented. For arm 3, where Mosin and Pola are given together, they need to be given on day 1 of each cycle. So if in cycle 3, day 1, Pola isn't given because of a certain toxicity, but Mosin is given, that dose of Pola is considered a missed dose. Again, we talked about the step up in Mosin. So if we have an interruption in Mosin dosing related to toxicity of 7 days or greater, it may be required that Mosin be given at the next or at the highest dose that was previously tolerated prior to resuming the planned escalation. In terms of Mosin related to CRS, that is all detailed in the protocol, and I have a slide on that in a couple slides. And then if treatment is delayed greater than 28 days, patients will need to be removed from the protocol. The local MD can contact the study chairs for an exception if they think it's necessitated. So I included dose modification rather interruptions for hematologic toxicity, because I think this is something that can be potentially fairly prevalent in the consolidation arms. So on day 1 of each cycle, the neutrophil count needs to be 1,000 or greater. The platelet count needs to be 75,000 or greater. If patients have grade 3 or 4 neutropenia, again, patients will necessitate growth factor on each subsequent cycle, and we recommend that growth factor is given prior to the subsequent cycle in order for those patients to be treated as soon as possible. Also recommend that CBC with DIF is checked frequently, and that patients are treated hopefully within 7 days or as soon as possible within that qualifying lab. The dose of Mosin and Pola is not going to be modified for neutropenia or thrombocytopenia. And if grade 3 or 4 neutropenia persists despite growth factor support, again, a local MD can contact the study chairs for consult. So I put this table up not to go over every detail, obviously, because CRS related to Mosin, it can be complex, but I just wanted the study teams to know that there are management guidelines for cytokine release syndrome related to Mosin in this protocol, including when to give anti-IL-6 therapy, when to give steroids, also what to do with the next dose of Mosin. And so those are all included in the protocol. And the same with seizure events related to Mosin. I don't anticipate that this is going to happen at the same prevalence as cytokine release syndrome may be, but this is also noted in the protocol in terms of management and what to do with the next dose. So criteria for removal from treatment for step 1 and step 2 and step 3. For step 1, if patients do not receive CAR T-cell therapy, or if they do not receive CAR T-cell within the window that has been laid out within 2 to 14 days of completion of LD chemo. If CAR T-cells did not meet those FDA requirements, again, if they receive it on a single patient IND or expanded access protocol, patients receive bridging and did not receive a PET scan after bridging, or if they receive polatuzumab or Mosin as part of their bridging therapy, they'll be removed from protocol. If they don't have a PET scan within 60 days of lymphodepleting chemotherapy, or if they don't have lymphodepleting chemotherapy within 60 days of registering, they'll be removed. And then for step 2 and step 3, completion of consolidation treatment, progressive disease, unacceptable toxicity, or any delay greater than 28 days, as was previously described. So I will pass this over to Kat, who will help.
So why don't we just take a little break, because that's a lot of information, and you can stay there.
Yeah, I think that's a good idea.
Just regarding the protocol itself, I think maybe you can frame, since you've had some experience giving Mosin Pola in the situation. You talked a lot about adverse events, but what you've actually seen as far as how straightforward that.
Yeah, so I think Mosin specifically, in terms of cytokine release syndrome, that's mitigated by the step-up dosing in cycle 1. And a lot of this is based on tumor burden for patients. At least that's what I found. I know a lot of other investigators have seen that as well. And with that step-up dosing, again, CRS is mitigated. And my expectation is that patients who are randomized to step 2 very likely will not have heavy tumor burden. So also have lymphodepleting chemotherapy previous and CAR T cell previous. So I think it's actually going to be very interesting to look at the CRS rates, but I'm really optimistic about those CRS rates being pretty minimal in step 2 for these step 2 randomized patients who are randomized to arms 1 and 3. Of course, we'll have to see what this is, but I think myself and a lot of the other investigators are certainly optimistic about that.
Just to emphasize for everyone, a lot of that requirements that Brian was discussing regarding the REMS criteria, that type of thing, that's all happening with these patients getting commercial CAR T. That's nothing that's special for this trial. Physicians that are signing patients up for commercial CAR T would have gone through all of that. That's really just pro forma that's in the protocol. So this is really ultimately designed as a pretty straightforward trial. For patient meets criteria for CAR T, they're likely to meet criteria for the study. And then it's a straightforward randomization.
Yeah, I think we tried to do our best when developing this study. CAR T patients can be complicated, but we did our best to try and ensure that everybody receiving CAR T, it was as close to standard of care as possible. So we really have large windows to make sure that we capture all of these patients. Again, for instance, we allow bridging therapy for all these patients as long as it's not Pola or Mosin. So I think the greater majority of patients receiving CD19 CAR T as standard of care are going to be eligible for this trial and good candidate for this trial. Does anybody have any questions about any of the logistics I went over, any of the eligibility criteria? Yeah.
Jeff Turn from Columbia. So if a patient's cell infusion is delayed more than 2 weeks beyond lymphodepletion, if they end up getting another round of lymphodepletion, can they still be eligible? Or?
Yeah. So those patients currently would not be eligible because they would receive lymphodepleting chemotherapy prior to. Now they could have another PET scan and likely be registered again. I'd have to talk to Huntley and Mike about that. It's the same thing for patients who that 60-day window from registration to starting LD chemotherapy. If you don't meet that window, for instance, if you have some significant toxicity due to bridging and that's delayed, you should be able to register again when you're outside of that window, but you still need to meet all those other parameters and have that repeat PET and everything. Any other questions? Yeah.
Because apparently there's some people on Zoom
Oh,
I have a question. Thanks.
Yeah.
It's on. Okay. Oh, hey, this is Jen from Columbia. Another question I had was, do you anticipate any issues with patients who might have cytopenias after CAR T? Because you do have, count restrictions, and it would still be in someone who's PR or stable disease as kind of standard of care, you might still be considering treating them with something. So I was wondering if you thought that might impact enrollment in any way.
yeah, I think that was one of the points that we tried to concentrate on the most. And that's one reason why we extended that randomization up until day 60. So if you have neutrophil count of less than 1,000 at day 30, we're seeing a lot of those counts improve by day 60. And you're also allowed to receive growth factor for those patients. It's not allowed to be received within 3 days of that qualifying blood draw. And the same thing for platelets too. That's been my experience for these patients. And we certainly have a number of patients that have those cytopenias at day 30 that would exclude them from step 2 randomization. And we'll be capturing that with the forms that Hung Lee has come out with. But I think by extending that window up to day 60, we should be able to capture the majority of those patients. There's no questions coming in through Zoom that I'm not aware of, I don't think.
Hi, I'm Kat Gassick. I'm the protocol project manager for lymphoma, and I will be helping with all the revisions coming forth for S2114. So when you are considering opening S2114 at your site, we wanted to list out 2 major considerations. One that Dr. Hess already covered, the risk evaluation and mitigation strategies associated with CAR T therapy, but that's all package insert information and isn't dictated by the protocol. The other important piece is the collection kits. So this is handled just like we do with all of our collection kits at SWOG with the SWOG Biospecimen Bank Management System. Please allow 5 to 7 days for shipment of those collection kits prior to each individual collection time point. So they will have to be ordered in advance of each collection time point. For drug ordering, we will be using McKesson, which we often work with McKesson as our distributor. If you order prior to 2 p.m. on any given day, they will ship it overnight, but please allow 5 days or 5 business days maximum in order to receive the shipment. You ought to receive all of the supply for each individual arm that patient is randomized to in a single shipment. And if you have any additional questions, as always, reach out to McKesson or please refer to sections 3.2 and 3.3 of the protocol.
Hello, everyone. My name is Laura Wells. I am a data coordinator with the SWOG Statistics and Data Management Center in Seattle. I'm just going to talk about registration timing. So for step 1, after consent and prior to LD chemo, LD chemo must start within 60 days of step 1 registration. Participants must receive CAR T cell infusion no later than 2 days and no earlier than 2 days and no later than 14 days after completion of the last day of LD chemotherapy. For step 2, after the day 30 PET and up to 60 days after CAR T infusion, patients must be randomized within 10 working days after receiving the email notification confirming the patient has either PR or SD at day 30 from central review, and after they meet all step 2 randomization eligibility criteria. Patients must plan to start consolidation no more than 14 calendar days after randomization. And then for step 3 patients, participants must be registered within 28 days from the date of progression. Participants must plan to start treatment no more than 21 calendar days from registration. And then a few data submission key points that can be found also in protocol section 14. All S2114 data will be submitted via metadata rave. Data must be submitted according to the protocol requirements for all participants registered, whether or not they are actually administered treatment, and including patients that aren't eligible. Participants who don't have documentation adequate to determine eligibility will generally be deemed ineligible. You can access the master form set via the 2114 protocol abstract page on CTSU website. You can see protocol section 14.4 for a more detailed data submission overview and time points. A few more key forms to be submitted. So for step 1, we'll have the on-study form, the CAR T cell therapy and lab values form, post-CAR T therapy and lab values form, the adverse event form, baseline and day 30 follow-up, tumor assessment form, and then all radiology and radiology reports will be uploaded to imaging via Triad to IROC. For CR patients and SD or PR, but who are ineligible for step 2 randomization, they'll need to submit a notice of intention not to randomize, as well as a follow-up tumor assessment form until progression, and another follow-up form. For step 2 randomization, they'll have another on-study form, treatment adverse events, and ICANS and CRS reporting form for all arms 1 through 3. And then arm 4 will have the observation notice form, and they will all have follow-up tumor assessment forms up until progression. Step 3 crossover will be the same CRFs as step 2 randomization. And they will have to submit a FUTA documenting progression before they move on to step 3. Okay, and some notes regarding image submission. They will have to digitally submit imaging at baseline and day 30. PET CT quality, they must be able to calculate standard SUVs. It has to go through local review. They must include SUV values for liver, mediastinum, and blood pool. They also must transfer the images and data through Triad, digital image submission, and central review. Eligibility review for step 2 randomization cannot be completed without both of the baseline and the day 30 PET submission. Rescans are allowed if the participant is still within the scan window. There's about a 4 to 5 day turnaround for the central review. And then IROC Ohio will notify the study PI, study statistician, and the sites of the results via email. And all direct questions on central review status will go to the IROC contact. And that's all I've got. That's it. Thank you.
Okay, so one other really important point of this study was we wanted to see if we can potentially refine who is high risk for relapse post CD19 CAR T with correlatives that we built into this study. And we know that minimal residual disease has shown to predict which patients are more likely to relapse post CAR T, but we don't have a lot of data on these specific subgroups and what it means for patients with stable disease or PR at day 30 or patients in CR at day 30. As I said, close to a third of these patients with CR at day 30 will be relapsing. So we built this into the protocol. We'll be collecting peripheral blood samples on all patients enrolled to step 1, with the first time point being prior to lymphodepleting chemotherapy. Again, all patients will have these collections at that day 30 PET scan, regardless of response. And similar to the disease assessments, these biomarker draws will be done relative to whether you're randomized or not, with the lab draws for 3 and 4 for partial remission and stable disease will be according to randomization, and CR will be according to CAR T infusion. The 5th time point will be for patients that have progressed. In terms of specimen submission, we have included here is the requirements for peripheral blood draw and tissue. All the peripheral blood will be collected in Streck tubes, and tissue, we're requiring that to be, the baseline tissue is for patients at diagnosis, or more preferably a relapse prior to CAR T. We're also asking for tissue at the time of progression, but this is optional. So does anybody have any questions about the peripheral blood draws related to the study? We just wanted to make it clear that these are for all patients that are enrolled to step 1 registration, and all the time points are clear, and that even if you have complete remission at that day 30 PET, you'll continue to have a couple biomarker draws that'll line up with those disease assessments. But happy to take any questions about any of those. Otherwise, we can move on to quality assurance. Yeah, go ahead.
This is just out of curiosity, but have we chosen the ctDNA assay yet?
No, we haven't chosen one specifically. We're planning on CAP-seq, so circulating tumor DNA for NGS. That's likely what we'll be doing. That's why we have these parameters here for 4 Streck tubes with 10 mLs of peripheral blood for each time point. So that's what we're planning. All right, Kat, for quality assurance. Sorry, I didn't have the order memorized.
So I'm Laura from Quality Assurance. So the audits are going to be part of treatment audit every 3 years. We'll come to your site and say hi and look through your notes and your documents and say, great job. So every 3 years. Is it this one? Yeah. Okay. Okay, so informed consent. So there's a single consent, and that's for the step 1 and step 2. And so we are recommending that, of course, you use patient-friendly plain language during the consent and the reconsent. And because this is a very complex study, we want you to make sure that you're reviewing the next steps with the patient at every study visit, which we know is probably being done anyway. And because there's a lag time between when you're registering the patient at screening and when they are starting treatment, we want to make sure that that conversation that the patient is willing to continue on is documented, because from quality assurance's perspective and from FDA's perspective and NCI's perspective, if there's a greater than 30-day window between when a patient is registered, consented and registered, and when they start treatment, there should be either a reconsent or a documented conversation that the patient is willing to continue on protocol. And so because there's only one consent and there's a big window, potentially a big window here, we want to make sure that sites understand that they need to be documenting that conversation, that the patient is willing to continue on treatment. So that's QA's recommendation.
Yeah, and that makes sense. And this is something we've kind of been talking about over the weeks leading up to this.
Yes.
I know we talked about potentially in the first revision, actually putting in that patients need to be reconsented.
Yes.
For step 2 randomization. I don't
Yes.
know, Hamli and Mike, do you guys have any opinions on that? Or Dr. Friedberg, I see you have an opinion.
So A couple of things. One is, I think you make a very important point about the lag time and that there has to be communication with the patient. You started by saying this was a highly complex study, and I actually don't want that to be the message out of this room.
Okay.
I don't
Okay.
think it's a highly complex study. This is standard treatment, and we have a straightforward randomization for patients who are in PR or stable disease at the end. It's really, there's not a bunch of decision points. Our colleagues in other diseases have much more complex studies than this. So
Okay.
it's a straightforward study, but you have this lag time. I think that the issue of reconsenting can get very confusing for patients because they've already signed, and the consent form is going to have language that isn't no longer relevant. So I think what traditionally has been done in these situations is that you in your note you remind the patient that they're on the study and that they're now going to be randomized, and that you have that documentation. But to ask the sites to do this, it's always hard because the consent form that's been approved is this initial consent form that speaks to the whole study. So unless you really break the consent form in half, where you have an initial consent form that only vaguely refers to what happens next, which probably isn't appropriate because the patient needs to know what they're getting into, I would vote for just one consent form, mainly for the sites, to make it easier for the site.
Okay. Well,
And remember, it's an easy, straightforward study.
yeah. Okay. Okay. I stand corrected. Thank you. Thank you for that feedback. But as long as sites are reminded that they surely need to be documenting that conversation, and we see it all the time when we're auditing, because we do give deficiencies when there is that greater than 30-day lag time and we don't see that documented conversation. And we tell y'all, we know you're talking to your patients. We know that you're having those conversations around consent. We know consent is an ongoing process. Write it down, document it. You know the old adage, if it ain't documented, it ain't done. So just make sure that you express the importance with your sites when you're giving this talk, or when people are reviewing this information, that there is a lag time between step 1 and step 2. Make sure that Mrs. Gonzalez is still willing to go on with the study, and she says yes, she wants to be treated. So thanks, y'all.
Do you have a question?
It's actually just a comment. I think that when you add the information into the protocol, and I think it's a good idea, just don't talk about reconsent. It's just a continuation of the consent process.
Okay.
Kind of as a continuation of the consent process, be sure that you document this conversation. If you talk about reconsent, it's a whole different thing, and you don't want to get into that.
Sure. Okay.
I'm Hildy Dillon. I'm the patient advocate that's worked with this study team from when it was just a pearl of an idea. And I will say, just to give you some perspective from the patient on this conversation that we've just been having, is that I agree with Jonathan, and with the study team, that in the study, it's not that the study is so complex from a clinical perspective, but we do need to keep in mind that patients are coming into CAR-T after they are pre-treated. They have consequences of that treatment that are both life impacting in terms of finances, and they're taking care of their families, plus the physical side effects of their disease and previous therapies. So it is important to have these conversations that you're talking about when you move on to step 2 for randomization, because patients are excited about the prospect and the opportunity for CAR T, but again, they're anxious at the same time. CAR T is not necessarily simple or symptom-free or side effect-free for these patients. And then we're talking about going on for more therapy, depending upon how they're randomized or if they've reached a CR or they progressed. So we had, for that, intentionally, I work with SWOG simple language specialist Amanda Stampless, who is here, on a simple language summary that you can use to go back to revisit with the patient at the various steps in the study so that they clearly understand what's coming next. A lot of the feedback that we get in advocacy is that people lose track of what's coming next in a trial. And it's really important in this trial because they will have gone through the rigor of CAR T before we're randomizing them, okay? This is available at the SWOG registration desk. I think, Kat, you were able to bring one here to this room. It will be uploaded to the CTSU website as a resource for you, as well as this clinical schema. This one is actually approved that you can share it with the patient. And we really highly recommend that you do that with every patient and then revisit it, you know, when they come in for those other visits. Also, the EMR template will be accessible from on the CTSU website. You go to documents, protocol-related documents, education and promotion filter on the protocol abstract page. And we have also worked on a financial barriers and resources for patients. We created this for another trial that we had at SWOG 1826 that is Metacruel. And I had several phone calls from site social work teams that it was very helpful to them. Remember, again, a lot of these patients have been through years of previous therapy. There are financial challenges in any family that's facing cancer for so many years. And this sheet provides resources from outside organizations external to SWOG that may be helpful for the healthcare providers to help their patients navigate out-of-pocket financial costs. Okay. Thank you.
Did you want to go ahead?
Hello, I'm Pat Mize. So this is a synopsis of the funding memo, and of note, the screening step actually has a higher amount of the base intervention payment because of the follow-up that's required for those patients who are not going on to step 2. Also of note, there's some additional funding per patient. And we also are covering the rescan for bridging therapy for PET CT scans. So that will be covered by the study. Is there any questions about funding?
Thanks, Pat. I think so. I think we just have a couple more slides left. I know we're running up against time, but just in terms of expected accrual for this study, again, assuming that about a third of patients at this day 30 PET will have stable disease or partial remission, and assuming that about 10% will drop out after first registration, we estimated we'll need 396 patients to register to step 1 to randomize 120 to step 2. So again, we've had a ton of cooperation from not only SWOG members, but ECOG and Alliance and BMT CTN. So I'm excited to open this up in a number of sites and have had a lot of people reach out to me, letting me know that they're going to be opening the trial. We're hoping to complete accrual within 24 months, in which we would need to register about 17 patients per month and randomize about 5 patients per month once we get a number of sites activated and open on the study. Now, I think, as Jonathan said, this is a really easy study to register patients on. All of my commercial CAR T patients for large cell lymphoma will be registered on this trial. The one exception is if they have a complete remission for some reason after bridging therapy. That's a very small percentage of these patients. So I really, estimate that we're going to be putting a lot of patients per year on this study at our center. Happy to take questions. If anybody has any questions about the study or about eligibility, please email the study team. But like I said, I think we did our best to make sure that there was no extra steps for these patients that are receiving FDA-approved standard of care CAR T-cell therapy for large cell lymphoma. So I definitely have to acknowledge all the people inside and outside of SWOG that worked to develop this study, to get it to where it's activated now. It's too many to name. Some people probably left off this list, but really been a group effort. And it's been really great to work with this group. I want to just call out Alicia and Kat and Kara and Hong Lee, who've just done an incredible amount of work on this study, more than probably nobody has worked more on this study than they have, I don't think. So thank you to all of you guys and everybody who contributed. And again, happy to take any questions. I won't go all over the forthcoming revision details in time, but thank you again. Mike and Hong Lee, do you guys have anything to say about the simple study? Yeah. That's why I'm going to start each presentation with. Yeah. We won't be getting, that'll be batched. So we'll be getting that after the study. Thanks for asking that. Yeah.
So between our institution, our cellular therapy group is separate from our lymphoma group. Any thoughts on how to handle that in terms of like where the protocol should sit? And we've already been going back and forth with our cellular therapy team for a month trying to figure it out. Because we feel like we're the ones treating them and making decisions on either of those times, but because they're getting apheresis and CAR T in the middle, there's some concern that they should be more involved. But I'm kind of curious if this has been an issue at all.
Yeah, I think that's a good question. At our center, they're combined. So my patients with lymphoma, they're progressing. I'm seeing them through CAR T and that, but all these patients are going to be getting standard of care CAR T. So you don't want them to, you want to consent these patients right after they're consented for CAR T collection. So do you want me to say that it should be you? Because I'm happy to say that.
I'm just curious if that's come up with any place else and how they might have handled it.
No, I haven't heard it, but it does need to be a collaboration. I think both groups need to believe in the study, and the cellular therapy group, again, just needs to understand what we've been saying, that these patients are going to get standard of care CD19 CAR T. So, and then when they come back, who's going to be giving these consolidation therapies? I think that could be a reasonable way to divide this. If you're going to be giving the consolidation therapies, then it makes sense for the stable disease PR patients. It makes sense for this to be with you guys. If those patients are going to stay with those cellular therapy teams, then, then that's, a potentially different story, I would say. But I'm also happy if, because a lot of this is promoted through the lymphoma group as well as the transplant cellular therapy group with Dr. Stiff. But I'm happy to talk at any of the cellular therapy CAR T meetings. If anybody needs more education, happy to do it. Well, guys, thanks for coming today. Really appreciate all the support.